First of all, good afternoon, and I would like to directly go into the presentation. My presentation is about the bioequivalence bioavailability study, what I have done. So generally the bioavailability means, which is the measurement of the extent of therapeutic active medicine that reaches the systemic circulation, where these are done with the test and reference where we come to know the bioequivalence. So the title of my study is Bioequivalence Study of Ritonavir Capsules 100 mg with Norvir Ritonavir Capsules soft gelatin 100 mg of Abbott Laboratories, which was the reference used as a reference product. I have done the study in adult male human subjects. Generally the studies for FDA would be done in fed and fasting conditions, but I have done in a fasting condition. Yeah, the background of the study is the present study was conducted to investigate the bioequivalence of Ritonavir capsules. It was of 100 mg. The methods and the patients used were generally healthy volunteers. It was an open label, balanced, randomized, 2 treatment, 2 period, 2 sequence, single dose, crossover pilot study, like the same volunteer was given a test and a reference. It was conducted in 12 healthy adult male subjects, age ranging from 18 to 45 years. Generally the FDA asks for 18 to 45 years, but if the molecule what we do requires, depending on the age, it can be altered. The total duration of the study was around 11 days. I mean, we can generally screen before. We generally don't include the screening period in the duration time because these are healthy volunteers. Whereas if you come to a patient study, it is strictly, I mean, once the screening starts, once he signs the consent, then starts the study period. I have considered 7 days as a washout period, considering the 5T half-life of the drug. The estimation of Ritonavir in human plasma is carried out by using the LCMS method in the bioanalytical laboratory with the help of my bioanalytical team, and also the statistical investigators, where the parameters assessed were the area under curve 0 to T, AUC, Cmax, Tmax. So the results obtained, the geometric mean ratios, generally which say the confidence interval should be 90% of the test and the reference drug for the test what we do. So for the Ritonavir, it was 100.8 for AUC, 102 for the AUC 0 to infinity, and 97 for the Cmax. So the 90% confidence intervals of test and reference was achieved, and the acceptance range for the bioequivalence. And the conclusion of the study was that the 2 Ritonavir capsules, the formulations which were done, the test and the reference products, were bioequivalent in terms of the rate and extent of absorption. So as per procedures, the pharmacist played an important role in the product selection decisions. They must generally, I mean, how the pharmacists play a critical role in dispensing. Even just before the guest lecture, Ms. Dhanlakshmi was explaining us clearly about the IP accountability and all. Generally, I mean, there are some definitions which I have put in my presentation, which talks about the relative and absolute bioavailability. If it is the next slide, it would be good. I mean, the bioavailability is a pharmacokinetic term that describes the rate and extent to which the active ingredient is absorbed from the drug product and becomes available at the drug action. So being this is a tablet, it would be the direct first metabolism where it goes to the liver and there the metabolism starts. Whereas the bioequivalences, 2 drugs are said to be bioequivalent where the rate and extent of absorption, which is the active ingredient of the drug products, were similar when administered at the same molar dose under similar conditions in an appropriately designed study. Well, the assessment of bioequivalence as per different regulatory authorities. For this, I have used a US regulatory. There are different kinds of studies what we do, pharmacokinetic, pharmacodynamic, clinical endpoint studies, but this is purely a BAB study, which is also called the pharmacokinetic. And the rationale of the study is when significant changes are made in the manufacture of the marketed formulation, when a new generic formulation is tested against the innovators or marketed product, it can either be bioequivalent or non-bioequivalent. Generally, if it is not, the product cannot be marketed. So the main objective of the study is to compare, as I said, and evaluate the single oral dose of Ritonavir 100 mg capsules. Coming to the hypothesis, I mean, it is the studies which generally talk about the safety and also the GCP. The approaches we make it to the study should be very done in an ethical conduct, where I have taken all the approvals from the regulatory and also from the ethics committee by submitting all the documents well prior 1 week, and I have attended the full core meeting and I've got the approval. So generally the bioequivalence study and design of the protocols happens through this way. I've given a flow diagram. So before commencing to the study, the protocol should be finalized, signed, and the ethics committee should approve. Pertaining to this, all the, I mean to say, not only the protocol, it should be the consent form, it should be the case report form, and all the volunteers should be explained clearly. And to be precise, even I have recorded some of the recordings for the study, what I have done by just taking their consent, but those should be kept confidential, and it should not be, it's only to the regulatory I should submit. And coming to the design and the methodology, coming to the design, it's a simple open label balanced randomized single dose 2 period two sequence 2 period. Generally the bioequivalence study should be conducted in a crossover, but only for few studies which have a long half-life and we consider when there is no safe, then we do a parallel design. If it is more than a month or so, we do a parallel design. Means one volunteer would be given test and the other volunteer would be reference, but by seeing the intravariability, the study, but for this it was a simple 12 healthy adult male subjects under 18 to 45 years. They were enrolled into the study. Considering this, the primary PK parameters were AUC 0 to T, the Cmax, and 0 to infinity. Whereas while doing, we also have secondary pharmacokinetic parameters, that is AU 0 to T, the Tmax. And coming to the investigational products, I have already said the products. The test is Ritonavir capsules. Whereas coming to the reference, I have taken Abbott Laboratories of USA. It is 100, both are 100 mg and both are capsules. It was done in oral. The inclusion criteria what I have used was, they were normally healthy between the age of 18 to 45 years. The both were included. The normal BMI range was 18.5 kilograms per meter square to 24.9, above 50 kgs. Basing on their health records, normal ECG, X-ray, everything, vital signs, their hematology and biochemistry parameters, and light smoke, taken non-smokers completely for this, and who are able to effectively communicate and adhere to the protocol requirements. Generally, the exclusion criteria, we say if they have any hypersensitivity generally to any drugs or food, we don't include them into the study, or whoever has been affected in the past with hepatitis or so, or any major illnesses and all, or if there was any significant history of alcoholism, because it would alter the metabolism of the drug, or presence of any drug abuse, or if they have donated any blood in the past, sure, the safety, or if they were. So the clinical phase procedure is this. The flowchart will explain us. I've done the screening. After that, the consent, then the check-in, dosing has been done. I've done in 2 periods. So after 7 days, they came back. After the checkout, the safety sample was taken, and we ensured that they were healthy. Then the bioanalytical process have been started. Basing on the concentration data, what we got, the PK statistical operations was performed, and generally we do like this. So me being for this PhD, I have just done, but I have did not submit it to the regulatory, but I have given the information to the ethics committee. So these are the ethical considered, the basic principles. The study was conducted in accordance with GCP, the Declaration of Helsinki, and So this was, as after taking the ethics committee approval, then the data analysis was done with this, and these were the pharmacokinetic and statistical phases. All this was performed. The results, coming to the results finally to conclude, the Tmax of the drug was somewhere around 3.25 hours and 4.5 for the reference product. The Cmax was almost similar to both of them. So these are the overall results what was done by the statistician. So the 90% of the CI intervals were within the normal ranges, which we say 80 to 125 range. So the summary and conclusion part. In the study, single oral dose Ritonavir capsules were compared to Norvir. In the study, the PK parameters of test formulation was compared with the reference formulation human beings from the individual time versus concentration data. The 90% confidence intervals of ratio estimates of Ritonavir capsules.